Hello, it's me again. So welcome back to uh welcome back to bio. Okay, so in the previous lesson we learned about the reducing sugar test, right? Which is called the Benedict test for reducing sugars. So now let's talk about complex carbohydrates. Okay, so what are complex carbohydrates? A complex carbohydrate is made up of many similar molecules of single sugars joined together to form a large molecule. Starch, glycogen, and cellulose are complex carbohydrates made up of numerous glucose molecules. The glucose molecules are linked in different ways, giving rise to different structures. Hence, starch, glycogen, and cellulose have different chemical and biological properties. Okay, so you can think of like glucose as like the basic building block, like maybe like a brick or something, which you can then use to build different structures. Let's talk about starch. So starch is a starch molecule is made up of several thousand glucose molecules joined together. Okay, over here you can see a molecule of starch. It's basically just like a long chain of glucose molecules. It's not all straight. Sometimes it's actually branched out a bit also. But yeah, in general, it's just a chain. Okay, so what's the function of the role of starch? It is a storage form of carbohydrates in plants. When needed, it can be digested to glucose to provide energy for cell activities. It is found in storage organs of plants, for example, potato, tubers, and tapioca. Okay, so basically, most root plants, right, like potatoes, carrots, everything that grows in the roots, they store the substances as starch. Okay, so yep, tapioca also. Yeah, tapioca is quite similar to potato if you've never tried it before. Okay, then let's talk about cellulose. A cellulose molecule is made up of many glucose molecules joined together. The bonds between glucose units are different from that in starch. The cellulose cell wall protects plant cells from bursting or damage. Cellulose cannot be digested in our intestines. They serve as dietary fiber that prevent constipation. They are present in the cell wall of plants. Okay, so cellulose is quite an interesting molecule because it is only found in plants, right? And the main function is to build the cell wall. Uh, Something interesting is that animals like cows, right, they actually can digest cellulose. They have the enzyme called cellulase, right? The ASE stands for the enzyme. And so the animals can digest cellulose, but humans, we don't have this cellulase, so we do not digest cellulose, right? It just passes through our system, our digestive system, and it comes out, in our feces. It's actually very important because if you don't eat enough uh, of this dietary fiber, right, then you will get constipation. Constipation is a symptom where your feces is too hard and it is not easily passed out from your body. It's like stuck there, that kind of feel. Okay, so yeah, cellulose is also similar to starch, right? It was just organized in a different way. Lastly, glycogen. Glycogen is a branch molecule. It is made up of many glucose molecules joined together. It is a storage form of carbohydrates in mammals. When needed, it is digested to glucose to provide energy for cell activity. It is stored in the liver and muscles of mammals. Okay, so glycogen, the last one. So you can see it's a chain of glucose molecules. This is inside you and this is inside of me. Alright, so all of these units are glucose molecules. So let's say that you just ate, like a big meal that's high in carbohydrates, high in sugars. Your body will then store the excess glucose as glycogens in your liver and in your muscles. And then let's say that it's been a while since you ate, maybe 5-6 hours. Now you're starting to feel hungry and there's no food for you, so your body will then break the glycogen back into glucose, and then you can use it for energy. Okay, let's talk a bit more about fiber. Okay, so yeah, basically they're saying here that fiber is important, and Yup. Fiber is important. Cool. Yeah. Okay. So let's move on. Okay, glycogen and starch are stores of glucose. Plants store glucose in the form of starch. Animals cannot make or store starch in their bodies. We get most of our carbohydrate from plants in the form of starch. Okay, so things like wheat, when we turn that into bread, things like rice is also a plant. Things like potato, that's also a plant. But we eat it, right? When we eat it, we are obtaining the starch. We are then breaking that starch down into glucose. Then in animals, the main store of glucose is glycogen. Hence, glycogen is sometimes called animal starch. Okay, that's a nickname. It's not a real name. Okay, it's just a nickname. So glycogen is stored in the liver and in the muscles. Okay, so let's see here. Starch is consumed and digested by animals, forming glucose, which is then stored in animals as glycogen. Okay. Glycogen and starch are suitable as storage materials in cells because they are insoluble in water, so they do not change the water potential in the cells. Okay, so unlike glucose, right, or sugars actually change water potential because when you have a high concentration of sugar, you lower the water potential. But because glycogen is insoluble, so it doesn't actually increase or decrease the water potential. It's just the same. So basically, it doesn't affect water potential. They are also large molecules, so they cannot diffuse through the cell membranes. They will not be lost from the cell. They cannot easily be broken down to glucose. They can be easily broken down to glucose when needed. For example, during tissue respiration. Okay, so we've learned a little bit about this thing called aerobic respiration. Right, this is where our body uses oxygen and glucose to release energy. Okay, there's also another form called anaerobic respiration, where our body doesn't need to use oxygen, but it just uses that glucose molecule. It breaks it, becomes lactic acid and a small amount of energy, and that happens when you are doing exercise that's very high energy demand at carb exercises. Okay, so lastly, their molecules have a compact shape, so they occupy less space than all the individual glucose molecules that make up a glycogen or starch molecule. Okay, so basically it's like, you in your house you have a storage room, right, where you store all your barang-barang, and you like just put all your stuff inside there. So that's kind of like the same concept. Okay, so let's talk about breakdown of starch. We have learned a bit about the enzymes in your Sec 2 chapters. So this is basically how the starch is broken down. Starch is in the food we eat and is digested in our alimentary canal. Okay, the word alimentary canal means your mouth to your anus. This pathway that all the way down, that's your alimentary canal. Okay, so when starch is digested, the bonds within the polysaccharide, okay, the word polysaccharide means many sugars, are broken and glucose molecules are released. The enzyme amylase digests starch to form the sugar maltose. Amylase cannot break maltose down to glucose. Maltose is hydrolyzed to glucose by another enzyme maltase. Note that complete digestion of starch gives glucose molecules. Okay, so the diagram. Okay, amylase is the enzyme that breaks down starch into maltose. Okay, why is it called amylase and not starchase or something like that? Okay, amylose is the scientific name for starch. Okay, so starch is actually just a common name. The scientific name is called amylose. So yeah, that's why it's called amylase. But don't worry, you don't have to call it amylose or amylase. You just call it starch. Okay, so alright, so now the maltase will then break down the maltose into glucose molecules. Okay, so that is how the starch is digested into glucose. Alright, now let's talk about fats. Alright, my favorite kind of nutrient. So what are fats? Fats learning outcome list the chemical elements that make up fat. State that lipids such as fats are synthesized from glycerol and fatty acids. State the main role of fats for insulation and long-term storage of energy in living organisms. Describe and carry out tests for fats using ethanol. Alright, so have you ever seen this, right? When you put your soup inside the fridge and then you see the oil harden into solids, right? These are your oil deposits and frozen oil or fats. Okay, so fats are a type of lipids. Okay, for the purposes of bio lesson, right, it's okay if you use it interchangeably, although it is not exactly the same, but it's quite similar. Alright, so fats are a type of lipid. So fats, like carbohydrates, are organic molecules made up of the elements carbon, hydrogen, and oxygen. Okay, you can just remember Mr. Choke, right? CHO and carbon, hydrogen, oxygen. It's the same as carbohydrate also. But unlike carbohydrates, fats contain much less oxygen in proportion to hydrogen. Okay, so there's a certain formula for calculating fats. Basically, the numbers can be very big, right? Much bigger than in the glucose molecules. And for example, beef fat has a formula of C57H110O6. So there's much lesser O. The proportion of O to C in carbohydrates is almost equal, whereas in this case it's much lesser. Okay, so yeah, you can see how little oxygen it contains in proportion of hydrogen. Okay, so basically the numbers are not fixed, right? It's just what, like, different fats got different numbers. Okay, so there's no need to memorize, just need to know that it's CHO. Alright, so fats can be animal fats or plant fats. Okay, I think it was a bit of a surprise for some people to realize that plants also produce fats. Okay, plants are not like purely green dietary fiber all the way. They also can actually convert their sugars into proteins and also can convert their sugars into fats also. Okay, so fats are commonly used as a store of energy, especially by animals. Let's look at this. Fats can be broken down into simpler compounds such as fatty acids and glycerol. Okay, this is a fat molecule, or you can even call it as a lipid molecule. The enzyme called lipase, right, will break it down into glycerol and 3 fatty acid molecules. Okay, so must write 3 fatty acid molecules. Yeah, okay, so this is one example here. This thing called tryptophan is a kind of lipid, right? Will be broken down by lipase into glycerol and 3 stearic acid molecules, which is one example of a fatty acid. But for us, we just know glycerol plus 3 fatty acid molecules. Okay, so the sources of fats are foods that are rich in fats include butter, cheese, fatty meat, olives, most nuts, peas, beans, seeds of castor oil, and palm oil. Okay, so this is why I'll say that the plants can also produce fats, right, by converting their sugars into oil or into the nuts. When they create nuts, the plant produces nuts, right? Then the nuts is actually filled with fats. The meat of most fishes and white meat have relatively less fats. However, some fishes such as herring and salmon have lots of fats. Okay, so the word white meat is used to describe meat that when you cut it, it looks whiter. There's not so much blood. So chicken, basically. Whereas red meat will be like beef, where when you cut the meat, you see a lot of blood come out. Okay, so yep. What are the functions of fats? Okay, so recently in Singapore, in this time, it's been a bit cold, so that's why I'm wearing a jacket even though the aircon is not on. I just open my window. Yeah, so it's very cold. and I'm very weak against cold because I don't have any fats. Right. So how are some animals able to survive in cold climates while others are not able to? What are the features common to these animals? How do these features help them survive in the cold? So fats function as a source and a long-term storage of energy. They are suitable long-term storage material because fats have a higher energy value compared to carbohydrates. Okay, what does that mean? It means that our body can store energy in the form of fats, because fats per gram, let's say like if you have 1 gram of fat versus 1 gram of carbohydrate, that 1 gram of fats can provide more energy than that gram of carbohydrates. Fats can be used as an insulating material that prevents excessive heat loss. For example, animals such as seals have a thick layer of fat called blubber beneath their skin, which helps to reduce loss of body heat. Okay, so just to show you, in case you never seen a seal, these are seals, right? You find them at the Antarctic. Antarctic is at the north or south, I don't quite remember. Antarctic. Okay, so it's at the bottom of the globe. Okay, so yeah, there was the seal. Other things like polar bears, right, also have a thick layer of fat to help reduce the loss of body heat, so they can stay warm for longer periods of time. Okay, fats is also a solvent for fat-soluble minerals and many other vital substances such as hormones. Okay, so not everything can dissolve in water. Some things like vitamins and hormones, they have to dissolve in fats. So the fats in our body will help to dissolve the vitamins and hormones. Fats is an essential part of cells, especially in the cell membrane. Okay, so like most of the cell membrane is made up of fats, and a little bit of the cell membrane is made up from proteins. Okay, so these proteins are used as channels to allow certain things to pass through. If you learn about active transport, then you will know that there's these protein molecules inside the cell membrane. And fats are a way to reduce water loss from the skin surface. Glands in the skin secrete an oily substance. The oily substance forms a thin layer over the skin surface, reducing the rate of evaporation of water, and the oily substance also reduces the rate of heat loss from the skin. Okay, so different animals, right, produce different things. So some animals, they are more like oily skin, some animals are less. Even for humans, we do have a thin layer of oil, right, like when we sweat and all that kind of thing, it does secrete a little bit of oil on our skin. Okay, but most of the time you feel uncomfortable with that, so you wipe it off your face. Okay, so yes, seal. This is a seal. This is the polar bear. Okay, so the test for fats is called the ethanol emulsion test. Okay, the word emulsion is important, so I'll explain what it means later. A white emulsion is formed when ethanol and fats are added to fats. What am I saying? A white emulsion is formed when ethanol and water are added to fats. An emulsion is a suspension of small drops of liquid in another liquid. Okay, so if you look at this picture of a white emulsion, these are actually small tiny droplets, right, inside another liquid. So that is why it's called as an emulsion. How do we test for fats using the ethanol emulsion test? There's two procedures. One is if you're working on liquids, and the other one is you're working on solids, but in general they both very similar. Okay, so in liquids we will add 2 cm cube of ethanol to a drop of liquid sample in a test tube and shake the mixture thoroughly. So ethanol is a specific type of alcohol, right? Similar to like the wine or the beer that people drink, that is a kind of alcohol. Okay, so in this case, ethanol, please don't drink it in the lab. Okay, yeah, it will do something when it mixes with the oil. Okay, so if you record observation, what you will see is this, right? A white emulsion. It'll be good to write. I've been taught to write a cloudy white emulsion is observed. Okay, then what happens if the food substance is not a liquid, it's a solid? For example, you have some peanuts or some beans or whatever, and you want to test it for oil or for fats. Then you will crush it using a pestle and mortar. Okay, so a pestle and mortar looks like this. Okay, this is pestle and mortar, so that you pound it. Okay, and then yeah, for this one, don't be careful. Some people they pound until their finger also gonna pound it. Okay, so yeah, basically they crush it, and then we need to remove the skin. So like, let's say in a peanut, got all the pieces of skin of the peanut, try and remove those. Anything that adds so much color or can't be crushed, then we need to remove. So we just crush the powder into a fine paste. Then we add it into a test tube, or put it into a test tube. Then we add 2 cm cube of ethanol and shake thoroughly. Okay, we need to shake thoroughly so that there's enough time for the ethanol to extract out the oils and the fats from the pieces of the sample. We will then allow the solid particles to settle. That means that like the peanut particles, right, because they are heavy, they will sink down to the bottom. Then we will carefully decant the ethanol, which means you pour off the top layer of the ethanol into another test tube containing 2 cm cube of water and shake the mixture. Okay, so what I try to do is you imagine that all of the peanut stuff is at the bottom. You are trying to just pour the things on top of the peanut into another test tube. So you don't want the peanut parts to move, you want the ethanol to move out. Okay, so once you pour over the ethanol into water, what you should see is a cloudy white emulsion. Okay, so yes, observation. The white emulsion produced indicates the presence of fats. Okay, please do the practice questions for yourself. Okay, now fats is often have a bad reputation, but actually fats is also important for our body. Okay, so this thing about eggs, right? Some people say, oh, don't eat the center, the yolk, the egg yolk, because it contains a lot of cholesterol. Yeah, it's true, right? Eggs naturally contain a lot of cholesterol because that's important for the baby chicken to grow. But as we learn about it, there's eggs. This cholesterol is actually useful in helping us to give energy to us and also build our cell membrane, etc., etc. But of course, eating too much of it then is negative. Okay, so everything in moderation. Okay, so let's now talk about proteins. Okay, what are proteins? Learning outcomes. These are the chemical elements that make up proteins. State that polypeptides and protein are synthesized from amino acids. State the main role of proteins for growth and repair of cells in living organisms. Describe and carry out tests for proteins using Burette solution. Okay, so people who don't eat meat are called vegetarians. So since they don't eat meat, they have to eat more dairy products, milk-based or soya products. This is because proteins are essential nutrients, and so alternative sources of protein must be consumed. Okay, if they don't eat enough protein, they start to lose muscle and they start to feel very weak. Proteins are organic molecules made up of the elements carbon, hydrogen, oxygen, and nitrogen. Okay, so the only difference here is that there's an addition of the word nitrogen as compared to your fats and your carbohydrates. So CHO and CHON. In some proteins, sulfur is present also. Okay, proteins are present in all cells. Their molecules are the largest and most complicated of all the food substances. Proteins are commonly found in the meat of animals, right? Fish, chicken, beef, etc. Okay, so in a protein is made up of many amino acids. And inside an amino acid, it contains 3 different parts. Okay, now it's getting a bit warm. Okay, so an amino acid is made up of 3 different sections. Firstly, it's made up of an amino group, which contains nitrogen and hydrogen, NH2. Over here, amino group. Okay, this amino group, there's different. Okay, no, yeah, basically just amino group. Okay, so there's also an acidic group, COOH, that's on the right hand side, and there's this thing called a side chain, which is sometimes containing sulfur. Okay, and so the general formula for amino acid is written like this. Okay, amino group, side chain, acidic group. So there are actually 20 different naturally occurring amino acids, and they differ because of their different side chain groups. Amino acids are combined in various ways to form millions of different protein molecules, just like the 26 letters of the alphabet can be used to form very large number of words. Okay, so all of these amino acids, right, they combine together to form different types of proteins. Okay, so long chains of amino acids fold to give protein a 3-dimensional shape. Many amino acid molecules are joined in a linear manner to form a polypeptide. Okay, so you can see amino acids for polypeptide. Okay, the word polypeptide means many, and the word poly means many, and peptide means these are peptides. Okay, so many peptides. Why? Because when you link an amino acid together, you have what you call as a peptide bond between them. So that's why it's called a polypeptide. Usually a polypeptide is like maybe anything like more than 2 amino acids already is called polypeptide. Okay, so like maybe 2 to about 10 amino acids you can consider as a polypeptide, but I think more than that usually is considered as a protein already. Okay, so the polypeptides are then linked up to form an even longer chain of amino acids, and a protein molecule is made up of one or more such chains folded together. In most proteins, the polypeptide chains are folded into a more complex 3-dimensional shape. Okay, so proteins are not 2D, they actually have a 3D shape, and that is the reason why enzymes, right, we've learned about enzymes, they actually have a specific shape for the enzymes to fit in. Okay, so let's look at some common enzyme 3D model. Okay, so this is how various enzymes look like in our body, right? They are represented by these models or these kind of blobs, and so yeah, that's how our body works. Okay, so summary: amino acids join to form polypeptides, which then join to form proteins. Okay, it also works the opposite way, right? So when we talk about digestion, proteins are digested into polypeptides in the stomach by this thing called trypsin, and then is then digested again from polypeptides into amino acids by various proteases. Okay, so sorry, a bit sleepy. Okay, what are the sources of proteins? Right, I don't think I need to go through this, but yeah, let's just say it. Milk, eggs, seafood, meat, chicken, lean beef, Then for plants, soya beans, nuts, grains, and vegetables, etc. Okay, what are the functions of proteins? Proteins are used in the synthesis of new cytoplasm, right, for growth and repair of worn out body cells, synthesis of enzymes and some hormones, and formation of antibodies to combat diseases. So yeah, we have talked about growth and repair, right? This is very important for proteins because the proteins are very important to make protoplasm, right? Oh yeah, cytoplasm and your protoplasm also. Your proteins are important to make enzymes. Majority of enzymes are actually made from proteins. Some hormones are made from protein, but others hormones can be made from other things also. Okay, so yeah, let's talk about the test for proteins. Okay, proteins can be detected by the Burette test. Okay, so same thing, right? It's invented by a guy probably called Burette. I don't really know for sure. Okay, the Burette solution is a blue solution made up of sodium hydroxide and copper 2 sulfate. Okay, so in different schools, some schools put it together already and give to students, but in Juyin actually is given separately. So this solution, Burette solution, is actually two different liquids that are put together, mixed together in specific amounts. Okay, you will see how to do it later. Okay, so this Burette solution, it will turn violet or deep purple when proteins are present. The test to test for proteins in a food sample, okay, one very simple one will be egg white. The reason why egg white is used is because egg white is transparent, so it doesn't really affect the color. If you use other substances that are like very colorful, like black color, like green, whatever color, that actually might prevent you from seeing a very clear violet color in the end, right? Because you get confused by the other colors or the other molecules that can change the color of the substance. Okay, so this is the shortcut version. We actually do the long method, but so how will we do it? We will take 2 cm cube of the liquid food sample. Okay, so we have the liquid food sample. Then we will add 2 cm cube of sodium hydroxide. Okay, as they say above here, sodium hydroxide. We will then add drops of, then we'll mix it, shake it up. We will then add copper 2 sulfate, which is what gives the blue color, right, into the mixture. We will shake it well. Okay, we add it like maybe drop by drop. So 1 drop shake, 2 drop shake, 3 drop shake, etc. Then we will shake it well and allow the mixture to stand for 5 minutes. Okay, actually it shouldn't even take 5 minutes. It'll be very fast. Within a few seconds you can see. Okay, so then we observe it. The reason why 5 minutes is just in case there's some like traces of protein. So like if there's really very little protein, then it may take some time for it to change color. Okay, so a violet color indicates the presence of proteins. The absence of proteins, the solution will remain blue. Okay, so in this case, it's not precipitate of anything. It's just solution turns violet, solution remains blue. Okay, can you try and do these practice questions? Okay, so yeah, let's just read this one and then we'll watch the videos for the Burette test. Okay, so how do we identify different nutrients present in our food? We will do various food tests, right? For example, on a peanut, cucumber, onion, tomato, banana. And we'll do this when we are doing the practical. Just quickly read this. Fats are bad for us. In short, okay, that's dumb. In order to obtain enough protein in our diet, we should eat more meat instead of plant-based food. You can get protein from plants also, but it's harder. Then, since the role of carbohydrates to what? Okay, these are all dumb. Never mind. Let's watch the video on Burette test.
Hello everyone. Today we are going to
Fast
discuss
forward
one of the
to
important reaction from
this part.
demonstration of biuret test. Hello everyone. So we have already seen theoretical aspects related to the biuret test, which is considered as the standard test for the protein. Now we will see procedure for the same. So first in the test tube take 3 ml of protein solution or test solution. In the test tube, take 3 ml of test solution. Then in the same test tube, add 2 ml of 5% NaOH.
NaOH is also known as sodium hydroxide.
Then add 2 to 3 drops of copper sulfate. And after adding it, mix it properly. You can see there is a development of violet color, that means protein is present in the solution. Now we will do control test. So for the control test, instead of test solution, you have to take 3 ml of distilled water. After taking 3
Okay, sorry about that, something wrong with the computer.
side. After adding it, mix it properly and then add 2 or 3 drops of copper sulfate and then mix it properly. So this is the control test for the biuret test.